show you right now what to do.
Okay.
We'll go through the practicalities of and revisit some of the theory in Galaxy. But here's your workflow, and you can see there's a lot of failed steps. This is from our group. And if you want to totally delete a history, you can do that. And you can go into here into saved histories. And this shows you a list of all your histories. And you can actually go delete permanently. If you delete, it will just be hidden. It's not fully deleted, just in case you want to undo. And no, I didn't really mean to delete that. But if you really mean to delete it, then delete permanently. It'll erase it off the disk, and it will free up space for others to do work. So if you weren't supposed to run a workflow, like we want everyone to run the workflow, but we just have limited disk space for everyone in the workshop to do it. So if you're not designated in your group, you should just delete permanently by, again, history options, saved histories, and delete permanently. If you want to delete an individual data set, you can delete individual data set by pressing on the X next to the data set, and that will delete it, but not purge it from the disk. In order to purge it from whatever's deleted but not purged has to be purged. So you go down to this option to purge deleted data sets. This won't necessarily remove an entire history of data sets. It's just the individual data sets that were deleted. Okay.
I'll put some screenshots of this in the Galaxy RNA Seq channel in Slack. So if somebody missed that, I can put it in there and you can see it.
Sounds great. So, sometimes it's useful when things fail, because I get to teach you we get to think about why it failed and fix it, because this is going to be a common experience for you. Experiments fail. I was told as a graduate student, if 10% of my experiments work, then I'm doing pretty well. Okay, so this is Catherine explained yesterday how a Galaxy workflow works, and we have these workflows that provide all the steps that you need, all the tools that you need in order with the correct settings to do the analysis that you want. And for us, what we were doing is trying to identify genes with statistically significant expression differences between two samples, okay? And I think everyone chose this workflow right here, which has a set of tools to do this. So when you get RNA-seq data, if you do RNA-seq experiments yourself, you're going to get FastQ files. So I just want to reiterate what FastQ files are, and look at FastQ file and what they mean. So we downloaded FastQ files from EBI, and you can, if you have FastQ files in your history, you can look at them with the view data, and you can see the format of these files. These are the files that come off the machine, and this first this is an entry right here. These 4 lines, every 4 lines is an entry for a read, and this entry for the first line contains information about that read. This is actually the machine, and some of these are the position on the slide that's imaged in the machine. So that's information about that read. This is the actual read of nucleotides. Then there's always a plus on this line. And then that's followed, as Catherine mentions, by an encoding of quality scores of whether that base was called correctly. So that's what these files look like, and they should look like that. And I believe the number sign, one of these symbols is a not good score. And if you end up seeing those, once you get used to these files, if you see a lot of, say, number signs or characters that you know mean it's a bad score, you should be concerned. But in general, I look through this and I see a lot of letters, and those letters for ASCII characters mean a really good score. So in general, the scores look pretty good for the FastQ files. It's formatted as I would expect. Okay, so FastQ files, and then what we did is we paired them up into the appropriate pairs. And you remember there was a left pair or forward and a reverse, and there was replicates. And so we did list of data set pairs. And then the first steps are really about making sure that these FastQ files have high quality reads. That's it. That's what all these FastQ type things, these different steps are making sure the FastQs are formatted correctly and are high quality. So the FastQ groomer was one tool we used, and it just actually changes this, makes sure that there's no spaces. It makes sure that the encoding is as you expect, etc. Okay, and that's a nice step. Basically all it does. It makes sure it's in the right format. And then, so FastQ groomer is a nice tool. FastQC is another tool that's really helpful, and it's useful to look at the output of that tool. That's what it's for, looking at the output. So you can see there's raw data and there's web page. And so I'm going to click on web page and I'm going to view it. And here's the results for the FastQC report. And it has all these categories that it checks to make sure all your reads are as expected. So we're going to scroll down, and the basic statistics, file name, file type, encoding, it actually figures out there's lots of different types of encoding for those quality scores. You remember those ASCII characters? There's a bunch of different versions, and it usually tries to guess what version it is, and it found that it was Sanger/Illumina 1.9. The total number of sequences, or the total number of reads that it identified, was I heard 21 million. So that's pretty good. Sequences flagged as poor quality or 0. Sequence length is 101. And usually you ask your sequencing core what sequencing length you want. You know, it's actually been increasing as the technology has improved with Illumina sequencing, and it gets over 100, but it used to be, you know, pretty low, like 40 or 50 reads, 50 nucleotides for each read. But this one happened to be 101. And the percent GC content, you would, you know, expect it to be 50, but this, I believe, is Plasmodium, so it has a high AT content, so GC is correspondingly low. And then the next thing we look at is the per base sequence quality, and this is these are all the positions across each read, and then on average, what is the score? Any score above 20, which is in negative log, that is a really good score. It's a really low probability that the base was called incorrectly. And you can see it's really high. It's in the green section, which is very good. And then it decreases over at the end of the read, and that's usually what happens. The longer read you have, the lower quality you get at the end of the read. But even at the end of the read, I would say the quality is very good. And then there's I'm just going to quickly mention these. The per tile sequence quality has to do with artifacts that have to do with the flow cell of the sequencing technology, and it identifies problems in each lane of the flow cell, and there doesn't seem to be any major problems. So the quality in general is really high across the reads in every lane of the flow cell. Per sequence quality scores, that looks good. Basically, the mean sequence quality for all reads is basically around 38, which is a really good score. Per base sequence content, you would expect the amount of A is equal to the amount of T, because they pair in DNA, and that usually is what you see. So the amount of A is the amount of T, and the amount of G is the amount of C. And that's good. If you don't see that, it's possible you're contaminated with RNA, which is not pairing, right? So you're not going to have an equal amount of T and A necessarily in RNA. Per sequence GC content, this usually is one peak. And if you end up seeing a bunch of peaks, that may mean you have contaminating stuff like RNA or even bacterial genomes or other things in there that have a different GC content. Okay, so this, you can read more about QC. I could go on for hours talking about this, but in general, the quality looks very good for this particular sample. There's no N's, so basically there are very few N's, which is the sequencer can't make a call. It can't make a base call. It doesn't know what base it is. So there's very few of that. That's good. The length of all the reads is 101, as we would expect, because maybe we asked our genome sequencing center and we paid for 101 nucleotide reads. Sequence duplication. This is actually pretty good. I'm not going to go into this, but I'm happy to talk about it more. Overrepresented sequences. There are a lot of times sequences that are not filtered out, like adapter sequences, RNAs, etc., and actually other contaminating sequences that are known to be there. And so these different algorithms look for those possible contaminating sequences. And then it looks for a lot of there's a lot of tools in this package for FastQC that looks for repetitive sequences. But we're actually expecting to see a lot of repeats because it's RNA-seq data, and there's a lot of genes that are highly expressed. And that means there's thousands of transcripts, and they're each sequenced many times. So in general, that's what the FastQC tells you, okay? The quality of your FastQ, your data and your reads. All right, that's good. But you notice that up at this graph above, that there are some reads, you see this huge error bar at the end. There's some reads that have, or especially at the ends of the read, that have very low base quality. Okay, we want to get rid of those because the sequencer wasn't very confident in making a base call. And so that's where we use Trim-O-Matic. And Trim-O-Matic just takes as an input the same data, the FastQ file, and its output is a FastQ file with the trimmed reads. It trims from the ends and it meets a certain specification. So on average, every 4 base pairs have to average a score of 20. So it trims the ends of the reads until that criteria is met. So we're still at FastQ, and now we have our FastQ, and we know that they look good from the FastQC report. We've trimmed poor quality bases from it. Now we're ready to do alignments. And this one failed, but I'll switch to another one. And here I'm at the high sat. And what this does is it's a short read alignment, and Illumina is considered short reads, even though it's 101. It's not like nanopore, which we saw earlier, which is, you know, sometimes 1,000 nucleotides or more. This is considered a short read aligner, and what we get is alignment to a genome, and this is where you have to specify what genome do you want to align to. And the result of this alignment is a BAM file. Okay, so that's the next file that we care about, is a BAM file. And we can look at, I think we can look at the BAM file. No, it downloads it. I'm gonna cancel that one. So just to let you know, here's the BAM file. That is an alignment file. It's a binary alignment file, and it is highly compressed as a file, and it's ordered by sequence. And you actually can look at those files. We're not doing that today, but I just want to let you know that you have access to this file as well, and you can look at it. You can download it here. This downloads the actual BAM file. You also need the index. And we did that. I did that with Gloria, another instructor today. And hopefully you can see this. Do you all see that?
Yes. Mm-hmm.
It's vector base and has all the aligned reads. This is one that was run last night. So we took the BAM file, we downloaded it with the index, and then we uploaded it to the vector base JBrowse, and we could look at it. And you know, we see there's a lot of alignments, and it's usually around genes. There's blue and red, so it's aligning to both strands. This is not a strand specific experiment, and I could sort of tell that because there's blue and red reads around this gene. And I would say it seemed to call introns a little too promiscuously. So maybe that's something to think about as we rerun this. Maybe we want to set a more stringent cutoff for when to call introns. But anyway, the point is that you have access to these files, and you can look at them and judge whether they're high quality. Then what we're getting, so we have the BAM file. That's the alignment file. And then the BAM file is really useful in two ways. One, converting it to a Bigwig. So converting it to a Bigwig, you've seen tracks that are Bigwig files, and that is the coverage track. So I can just look for alignment here. I could clear this. Why am I not seeing it? It must be called something else here. Anyway, do you remember that histogram that tells you how many reads map to that location? So that's what a Bigwig file is. You've seen it already. And here is one example. Just not showing up. Let me zoom out in case there's no expression here in this location. And that is indeed the case. So this is a Bigwig file. I take the BAM file, and then it looks at every position and figures out how many reads overlap that position. And that's where you get the height of that histogram. So that tells you where all the reads in general are mapping. Okay, so that is a Bigwig file. You can actually even look at this save track data. Actually, that's another form, a bed graph, but it's very similar in producing a histogram of the reads. So that's why we did that Bigwig step, and we're going to import in your in your breakout rooms after this, we're going to look at the Bigwig files. We're going to move them to vector-based or plasmodb and look at those particular files. So that's one type of file that's generated from a BAM file. So BAM from the HiSat to a Bigwig. And now BAM is also used in HTC count. And that's a very specific tool for RNA-seq analysis. And what that does is it looks at all the reads and it tries to identify those reads that map to parts of genes. You specify this. Does it map to the whole gene? Does it map to an exon? You specified this in the workflow, and it counts the number of reads that map to each gene. And that's representative of the expression of that mRNA. So let's just look at that HTC count, one of those. I'm going to first go to this data set, view details, and what you did is selected a feature type of exon, so it counts all of the reads that map to exons. And then this ID attribute, it uses this to sum all exons based on the gene ID. So if you have gene A and gene B, and each has 3 exons, it will add all the 3 exons for gene A, and then add all the 3 exons for gene B. And it will report 2 numbers, one for gene A, exons, and one for gene B, exons. So that's what we're getting. So let's just take a look at one of those files. It looks like this. It's just a simple tab delimited file. Here's the gene. This is the gene ID. And here are all the counts that map to that gene. Not that gene anywhere, but specifically to the exons of that gene. Okay. And you can specify whatever you want. It could map to anywhere on the gene if you want. You could change those parameters to do that. But we chose exon. That's usually the accepted way to analyze RNA-seq data. So HTC count, that gives you the counts, the number of reads that map to that gene. And then that can be fed into DE-seq too. And that's a really great statistical package that allows you to analyze count data for RNA-seq. And it figures out, is there a significant change in the number of reads for each gene? So we'll look at DE-seq 2. We'll first look at this file, the results file that you'll look at. And here's the gene. This is the number of reads across all the samples. This is actually the fold change between your two. You know how you set up sample A versus sample B. This is the fold change in log2 space. So it went down 2 to the 14th time. I don't know that number, but 2, 4, 8, 16, 32, 64, 128, a lot. It went millions. That's how much it changed. And then this is the standard deviation of that change. And then this is a statistic that it used. It's sort of like a chi-square. It's called the Wald statistic. And then it uses that statistic to figure out the p-value of that change. That's a really low p-value. And you've heard about multiple hypothesis testing, because we're doing this test for every single gene, thousands of genes. So we need to correct for that. So this is the corrected p-value that we care about. And that's still a very significant change. So the 2 numbers I usually focus on are this fold change and then this p-value. And that gives me an idea of the significantly changed genes. There's There's another way to look at the DE-seq2 results as a whole. You may wonder what's going on with all the genes as a whole. And this set of plots is really useful. I'm just going to focus on this plot right here. So this is mean expression. These are really lowly expressed genes. These are highly expressed genes. And this is the fold change. Is this gene going up or is this gene going down? And in red are the ones that are significantly changed. So you can see that the bigger change there is, the more significant they are. But as you get to higher expressed genes, you don't need that much of a fold increase or fold decrease to get a significant change, because the reads are so much that it aids the statistics. Okay, so that is basically all the files that you're looking at. What we're going to be doing is exporting 2 sets of files to PlasmoDB or Vectorbase to actually analyze them there. And your group will help you do that. So you're going to be exporting the Bigwig files that I said were really important to look at, and also the HT-seq data. You're going to convert that to a normalized expression value and then export that to our website to explore. So any other questions before we go into breakout rooms to deal with this stuff?
The HT-seq, no, he aligns the sequences to the genome. You probably know that in HT-seq, can I adjust the parameters so that it marks reads that are in the upstream region of genes? And can you separate that, the routine?
That's a good question. So you can actually set any region you want as long as it's in the GFF file. So in a GFF file, usually it's just for genes, and it has the whole gene, the start and the end of the gene, and it has where the exons are, where the CDSs are, where the introns are, where the UTRs are at, and usually that's it. If it's in there, you can make HT-seq count that. And for what you're talking about, looking for reads, like quantitating reads that are in the promoter or upstream region, people sometimes make their own GFF files and use that for HT-seq analysis and call that region in the GFF file upstream or something, and then specify in the parameter for HT-seq, which is, I'll show you. This the feature type, what do you want to count? It would be your upstream, whatever you name it in the GFF file. And you can do that. Sometimes people use a tiling, sort of a tiling, like, I want to know the quantity for every 50 nucleotides across the whole genome. You could do that. You could make a GFF file like that and use each HT-seq in that way.
Okay. No, I was just thinking about how to analyze ChIP-seq data. So
ChIP-seq, we were not talking about that here, but for ChIP-seq, you're going to do the same thing with the FastQ files. You're going to align to the genome with like HiSat or TopHat or some other aligner, and then you're going to diverge and use some other tools to help you identify peaks. So this is not the best place. HT-seq is not the best tool for ChIP-seq. A peak calling algorithm, there's lots of them. We might have them here. peak calling. See the peak call, all these tools, what they
Yeah.
do is look in the BAM files and they try to identify peaks in all the reads.
And Mark,
So
the first thing you're going to have to do, though, I mean, obviously after trimming, is aligning to the genome. So you're going to have to align them using some program, whether it's HiSat2 or something else,
Yeah.
to generate a BAM file.
Right. You'll have a BAM file, an alignment file, and then you'll feed it to one of these things.
Yeah.
Okay. Great.
Are there
So-
other questions from anyone? I know there was a lot of issues with Galaxy workflows last night, and Omar sort of described what might be the issues that cause that. So I can spend a little bit more time if you have specific questions before we break out, so it can be
So
more
what we're-
productive.
Yeah, maybe just to what we're going to do is, because I'm pretty sure almost every group, and we'll find out when we go into the breakout sessions for sure. I think everybody had some issues, and most likely probably had to do, I'm guessing, had to do with a memory issue. So what we did is we pre-ran a bunch of the workflows. There are a couple of them that didn't, unfortunately, that failed even for us before. So we'll just substitute those with other ones. So when we break out, depending which group you are, we will share with you the correct link, which will allow you then to import the workflow into your workspace and then work with it. And we can do that in our groups. We should be able to do this on an individual basis. If things slow down dramatically, then we'll know that we're maybe even approaching another limit of space, and we'll have to adjust accordingly. So I think, unless there are any other questions, we should probably go into our breakout rooms and sort things within the breakout rooms. Mark or Catherine or other instructors, any other issues?
Groups 1, 2, and 3, we have, for that Plasmodium burgii data set, we have group 2 workflow finished, and I've added that link into the Slack channel. So I think all 3 of those groups are going to need to analyze the results of group 2.
Yep, I'm assuming groups 1, 2, and 3, they all failed, right? They all had problems as well. So
Yeah, we'll see.
okay, we'll put the links somewhere in the chat or in the Slack, so you will get them in a second. Okay, let's go to breakouts.
See you soon.